step 4, which is droplet reading and counting. Now it is important. You have to, because what we need, droplet quantification by absolute number. So it is very much similar to a flow-based system where there the cells are used with sheet fluid in the single file, so that you can take one cell at a time, forward and side scatter. You can write them like in here also. Droplets are used in a single file. So it analyzes and counts. Analyzes also, positive, negative, and then counts also the numbers. For positive, negative, using two color detector system, whether it might be PAM or positive or X or BIC, which is a proprietary control. So by using that, not just we can use one or two things, more than one or two, or multiplexing. So multiplexing is also one of the interesting aspects that it provides, that multiple sample, multiple targets you can identify or analyze at one time. Next slide. Now, coming to counting, so you see on the leftmost microcentrifuge tube where you have a reaction mixture, then these are droplets. After that, when you analyze them, what might be the scenario? It's, you are taking as sir was saying, liquid biopsy. There can be wild type cell or some mutant cell. So in that case, you can have some droplet having only wild type or the background DNA, then your target of DNA of interest. The rest of might be empty as well, empty over there. Then now you have to calculate what the fraction of positive droplet. That is fairly easy. Positive droplet divided by the total number of droplet that you have. So, and after that you will apply Poisson's statistics. It is done by the computer or the software itself, which helps you to identify absolute copy number. Now a question arises here that if you are counting cells and if you are not cells, your droplets, and you can identify based on fluorescence positive, negative, then why do we need this Poisson statistics, which is complex physics-based application? Because consider three scenarios. I'll be coming to them. Low copy number or low sample positivity, intermediate and high. What is there? High sample positivity. Suppose you have a 100 glasses, cup size glasses there, and I have 10 grains of rice. I am putting there. What are the probability that one single cup will have more than one rice? Very low. If I am having 5,000 rice grains, then the probability of having more than one grain in a cup is more. So this is how this assumption or statistics actually let you know that whether the fluorescence is due to a single DNA target or more than one. So in that way you can count that. If it is more than one, you can always count, take it as 2 or 3. So that is why we need Poisson statistics. Now this is how the result of ddPCR looks like. You have this background red line above which you have the positive droplets scored as 1 and negative droplets scored 0. We do not score more than 1 because 2 nahi hai, because we do not know if the fluorescence is there. It is due to one copy of the DNA or more than one. So that is the reason why we need this Poisson's statistics. So this is how the result of digital PCR looks like, where we plot fluorescence intensity versus your droplet count. Now, like I said, for Poisson's modeling, what sample is randomly distributed. So one can have one or more tablet, and one droplet can have no tablet at all. So this is the formula that we propose. This is coefficient per droplet given by negative logarithm 1 minus p, where p is a fraction of positive droplet. you know how to calculate positive droplets. Like here you can see some empty droplets, some having two targets, some having one only, some having negative. So in this way, this Poisson statistics will have this way. So he was the guy of Poisson, the scientist. He was a French physicist and mathematician. I hope you might have heard about this in elasticity, Poisson's correlation, and these things. So he did this determines the probability of a micro reactor to have more than one, as I said, copies of original target. So assumption low concentration, as I said, most of the particles like let's say 150 droplets are there, only 6 are positive. So most of the particles will contain will be empty, and only few will be having one copy. And it's a very rare chances of having two or more than two, because given this is a very low concentration. And at high concentration, you'll be having more than one cup copy, because I explained with this example of the cup and the rice, like that. Next thing. So again, just to show the questions, your statistics at very low. Lambda is your concentration, you can say, sample concentration. Then, as the sample concentration is very low, the empty your droplets, that is zero, is maximum. Then, as the sample concentration increases, the droplets having more than 1, 2, 3, even 5, 6, 7, they are there. Okay. Next slide. So this is the correlation given for high occupancy, this formula. So don't go, if you take my advice in these formulas, just let the software do the work for you. Okay. Come to the next slide. Again the same thing, no targets, low targets, and here you have one, let's say 143 droplets, and low concentration we have 6 on 143, but using that Poisson statistics, the corrected counts come to be around 6.2 in the low, just similar to that. And in the medium concentration, around 30, 34 is there. If you apply Poisson statistics, it will give the corrected value as 38. Whereas in high concentration, you see 70 are there, but on correction, it gave you a high value of 96. So this is how the Poisson statistics help you to quantitate the absolute numbers. Now there is one more term that is applied for digital PCR, that is CPD, that is copies per droplet. Very simple copy, how many copies in a droplet. Okay. So again, in the same scenario, I'm explaining the same thing. 6 copies in 20 microliter sample. So we will expect to find exactly 6 droplets that contain tablet, because it is very unlikely to have the scenario that will have more than one. Then at next slide, intermediate concentration, where in some cases it can have up to 3 or 4 target DNA copies. Like assume 5,000 targets in 20,000 droplets. So if you go mathematically, 75% of the droplet should be empty, but as per the Poisson's distribution, it's not 75, 78%, because some of the droplet might be containing more than one. So that is why not 25. The positive, the number of positive droplets will be slightly less than expected, because occupancy, the positive occupancy is more per droplets. And in high concentration, let's say 50,000 target molecule, and the copy number is very high. It's some samples like that. So 50,000, let's assume in 20,000. So around one droplet will have around more than 2.5. So what you can assume? Will there be any empty droplets considering these assumptions? Because they are highly populated, these targeting, but Poisson predicts this software calculus around 1642 will be your empty droplets also. So this is based on that software, the snapshot in 50,000 you have 1642 will be having zero droplets, then even you have 10, 11 or 9 droplets that can be calculated. And that way you can count the absolute numbers because you have the positive droplet numbers, but the numbers will be more as the target DNA samples will be having more. Next slide. Now, coming to the benefits. First and foremost, why we are doing this? Because we want something more than RT-PCR, real-time PCR, that absolute target quantification that can be done. Then it has high tolerance to inhibitors, or there is no competition for reagents because each and every your microspheres or the droplet is an independent reactor. So inside the reactor there can't be any competition and they are isolated, segregated, discrete from others. So no competition and they are having high tolerance to inhibitors like heparin and other inhibitors that tend to affect your tag polymerase and other things in the qPCR. And its precision is also high and sensitivity and then multiplexing. Multiplexing we can do because we can increase the number of fluorophores and there can be multiple permutation combinations also. And high reproducibility. Because in your real-time PCR, you repeat your experiment. People say that, yeah, they have a great reproducibility, but trust me, reproducibility is a problem there. You can have different CTs every time, but here, if you have an automated droplet generator, the reproducibility is excellent. Only the problem is cost, as sir was also saying, but also the cost is high. Now, limitations. The most important advantage was uniform size of droplet for your quantification, and most important limitation also becomes variable in the size of the droplet. Okay. And as droplet generator and analyzer, then your things are multiple instruments are needed. So time consuming, obviously it is a time consuming thing, and so many pipeline, oil and droplet and other things are there, proprietary chemicals are there. So there is risk, obviously risk of cross contamination. Now coming to application, quantitative analysis as in copy number variation. Like in some cases, the copy number differences are very subtle, like 1 or 2 or 3. How can you rely on your real-time PCR because they can be, non-manifested in the real-time PCR because they can be embedded in the noise, or you will not have proper CT value. Why? In the case of digital droplet PCR, as you have multiple droplets, you can find your positive. And so for various diseases, for variations are there, it can be used like psoriasis, Williams syndrome, and schizophrenia, and so on and so forth. Now, it is rare mutation. It is very rare in then you can use this for because of the reaction partitioning effect, effectively increase the rare target detection. So this is problematic in case of real-time PCR. Then coming to the next slide. And liquid biopsies, and I don't need to talk about liquid biopsies anymore. I think Anand sir has so beautifully told you for that. So here also you can do your use this ddPCR for multiple things. You take out the DNA, you use process generator, you use multiple fluorophores and the various permutation combination. As I said, you can help take the help of liquid biopsies. ddPCR and liquid biopsies for diagnosis, prognosis, recurrence, disease monitoring. Next thing. Now coming to the thing that I have worked, genome editing detection. The CRISPR-Cas9 system, which is a very popular method and cost-effective method to edit the gene, where you use what you use here? Guide RNAs. Guide RNAs, single or multiple or tandem guide RNAs. If you use single guide RNA, error-prone your it will introduce a non-homologous enzyme, and if you use double or tandem guide RNA, then that will bring about long or large segment deletion. So let's say how do you do in the cell lines? You have a cell line, let's say glioblastoma cell line, I got it. And after that you have your CRISPR construct where you are incorporating those guide RNA in your vector and you transfect them in the eukaryotic cell culture. And after transfection, what do you expect? Do you expect all the cells to take the guide RNAs? No. Or the vector itself? No. Then it becomes really difficult if you want to check for your deletion initially because you have to wait. These vectors will have what? For selection, we have puromycin resistant gene. So you have to grow those cells in puromycin selection over a period of time. Initially, what will happen? Few untransfected cells will die, some less resistant cells will die. You want only cells that are positive. But what can happen? Because I encountered it. I got my clones, but there was no deletion. Why? Some cells are also inherently resistant to puromycin. So in that case, I had a really difficult time because I had to screen for multiple clones and luckily only one or two were positive. Otherwise, they all were resistant. So consider this as a major hurdle. So how can you use this ddPCR? In this, what you can do, you have your cell line, you have transfected them. After few days, what you can do, you use those cells and for droplet generation. And depending on the primers that you design for either deletion or your the long segment deletion or mutation, you can identify which subsets of the cell are having truly genomic editing has been taken place. So what you can do, you can flow sort those cells where you have long segment deletion or you can have only indel mutations. So in that way, you can get your clones easily and early. While after doing your after showing your knockout by Western blot, you also need to validate that by PCR whether there is actually indeed any deletion or mutation or not. So by this digital PCR, you can save the time. Next. Then also gene expression analysis when there is a very minor fold chain differences, obviously. Next slide. And it is used for viral load measurement. In US they use for detecting the level of SARS-CoV-2 in the water samples. So it was not FDA approved, it was emergency users license approved. So it was used in this case also. Then environmental monitoring where you can you want to detect specific microorganisms or environmental samples you want to analyze. So it can help in research. Next slide, please. NIPT can use obviously as a digital ddPCR to detect the fetal chromosomal anomaly from your blood sample. Next slide. Then monitoring minimal residual disease after chemotherapy, after radiotherapy, you want to see, like I said, cancer stem cells or circulating tumor cells. Then you can use this for PCR amplification using digital PCR, and you can also count the number of positive events. Next slide. So to summarize, digital droplet PCR was delivery and precision in various scientific disciplines, healthcare, research, disease management, even your environmental and biotechnology also. And the main crux or the main advantage is that it provides you means to absolute quantification of your nucleic acid, so that it will help to in various cases where real-time PCR is not good enough, or that you have to go for NGS, which is far more expensive. So that is a valid enough option. Next. But there are limitations also. First, you have high cost per sample. Then droplet generation, as I said, if you are not good at this stage, the whole service is lost. Then equipment is there and not therefore ideal for us. So in nutshell, what are the benefits? Absolute target quantification, high tolerance to inhibitors, precision is high, sensitivity is high, reproducibility is also high. And at the same time, what are the applications? Copy number variation, certain changes in copy number, pair mutation, library entries, library quantification, viral load, environmental things, unit pressure analysis, etc. So, what are the various future directions in DDPCR? So we can go for multiplexing, that will be really good. In fact, multiplexing is done, but it should be the main aim. Then for POCT platforms, the DDPCR, it can be used. Then for various diagnostics in an automated manner. So that's all. Thank you. Thank you for listening. And if you have any questions, please.
ये पूरा सारा तुमको निकाल दिया
Any questions? Yes, please.
माइक
Yeah, that was a very nice presentation. You mentioned about one of the limitations is the droplet generation errors, right? So both it's saying and claim that the generator generates around 20,000 droplets. But in reality, what are the actual acceptable droplets that are acceptable before or taking it positive for? Do you actually get 20,000 or you get it less than that?
You get that.
You get every time 20,000 droplets?
Because at nano level, if we just by looking at the number, the volume wise, around 10 to 20,000, 20,000, 15 to 20,000, and 80 or 90% of the droplet should be ideally of the size, optimal size.
Okay, yeah, thank you.
बैठो तुम बैठो ना ले लेगा वो कर देगा
Thank you, Dr. Anurag. My experience, here could you place a digital over RT-PCR or conventional PCR, whether how it adds clinical value to the diagnosis or to the relapse, and how should the laboratories strategically plan with this, with one of these PCRs?
Actually, if you look at the advantages that we had with qPCR, that's robust advantage. They are cheap, less time consuming, and for a variety of applications. But the limitations of qPCR becomes the catching point of DDPCR. Because if there is very low copy differences or very low frequency mutations, that becomes difficult. How can you quantitate those things? Because the qPCR will generate so much of noise. You have to move you have to play with the threshold, so it becomes unpredictable. So in those cases, as it deals with absolute quantification, if they are even in very less amount, yes, you can have that. And if you add multiplexing, well, it is even better.
Yeah, my question is in terms of handling of the instrument part, like when we are doing digital PCR or droplet digital PCR and on one side your conventional PCRs, what are the operational challenges which are there, like in terms of why I'm asking this question is like if we want to use it more frequently, it has to be made more user friendly. So in terms of operational challenges, what are the key areas which are involved?
If you see, all set of consumables are different, the pipettes are different. Then you have to pipette multiple times. So first and foremost, this is the challenge. Then you are dealing with the proprietary chemicals, so you have to handle it with care, always, because it's very expensive. And operational challenges, as I have also not been exposed to this. So theoretically, I can say that handling and droplet generation remains major challenge. So by having proper guidance or experience only, we can overcome this. So I am also very new to this, but yes, these are the obvious challenges.
Thank you.
What is the cost?
Cost, it may on an average 20-25,000 at least. But Sir Arnav sir was saying they are planning some of the ways to cut it down. But yes, please.
For particularly low concentration samples. Measurable after chemotherapy or so. How this digital PCR is superior to the RT PCR? Because here we measure the absolute concentration. So in case of low concentration samples, if the signals, as we know that in the data PCR, the signals are sum up of each value. Like if it is lower than a certain intensity, it will be negative, and higher than that, it is positive. So high concentration sample is okay, but for low concentration sample, how it is superior to RT-PCR?
The RT-PCR, you are talking about real-time PCR.
Yeah, it's a
Okay.
we can examine that all these values we need to detect. And we are in the because it's done absolute. Then not different compared to artificial to detect in low concentration compared to artificial.
As of my experience, anything that is having low incidence or low copy number, it becomes difficult because we are not only working with experienced students, experienced faculty and scientists. There are so many students also. So for them, to get to the CT value and to differentiate whether it is noise or whether it is true signal, that is little different, difficult in real time. PCR. Why? Here you don't have to worry about CT or standards, not at all. Just you have to see and rely on the machine and they will count and tell you if the target is there. This is what I think. Any more questions? So thank you so much. Thank you for hearing.
Thank you, sir, for the comprehensive and highly engaging session. Your insights have truly enriched our understanding and added immense value to today's event. It is my privilege to introduce our next distinguished speaker, Professor Dr. Swaroop A.V. Shah, sir, one of the leading molecular scientists and a prominent voice in the field of human genetics and precision diagnostics. Serves as a senior molecular scientist in human genetics at the Department of Laboratory Medicine and Biochemistry, PD Hinduja National Hospital and Medical Research Center, Mumbai. A PhD scholar and a fellow of the Academy of Diagnostics and Laboratory Medicine, Sir has built an impressive scientific career at the intersection of molecular biology, clinical biochemistry, and genomic medicine. With multiple publications and citations, his research portfolio reflects both depth and influence. He is a member of the IFCC Emerging Technologies Division, and at Hinduja, he is recognized as a PhD mentor, a principal investigator on multiple funded projects, and a respected leader in translational research. Today, he will enlighten us with his talk, Integrating Molecular Diagnostics into Precision Medicine, from bench to bedside. Ladies and gentlemen, please join me in welcoming Dr. Swaroop A.V. Shah, sir.
Thank you for such a lovely introduction. We're just 1 minute before it starts. Good afternoon. So, good morning to everyone. So, firstly, I would like to thank the organizing committee, especially Dr. Swarnima, for the kind invitation to be a part of this excellent summit. And this is my first time in Patna, and I'm really having a good time amongst you all in such a scientific feast. So, to begin, I'm going to talk about how molecular diagnostics will play an important role in precision medicine and from translating from bench to bedside. But before I begin, this is the place where I work and I've been associated for almost 2 decades now, which is PD Individual Hospital and Medical Research Center. It's one of the leading private hospital in the country, and okay. Okay, so it was established way back in the 1950s. We are more than 500 bedded hospital. We treat around 1 lakh patients annually, and we perform something like 11 lakhs laboratory investigations. We also have a mobile health unit which caters to 47 rural villages in Maharashtra. And we were also the first hospital laboratory in the entire South nations to be accredited by the College of American Pathologists way back in 2006. Okay, next slide. I think this is okay. Okay, this way it goes. Okay, so this is going to be my presentation outline. I will be talking about the concept of molecular diagnostics. We'll touch upon how important is precision medicine, what are the adverse drug reactions, the role of predictive biomarkers in that, in pharmacogenomics, what are the resources and guidelines that are available, what are the barriers for its effective implementation, and I would be talking about our own hospital-based study, which is a classic example of how research from the lab was translated into clinics. And of course, I will be end with future directions in precision medicine. So, molecular diagnostics. Okay, I'm sure you all know about this, but I will just quickly refresh our understanding of the basics of genetics. We humans are made up of billions of cells, and each nucleated cell contains a genome, which basically are the chromosomes that we inherit from our parents. These chromosomes are nothing but supercoiled DNAs, which are made up of 4 nucleotides that contains information in the form of genes. And this DNA is then transcribed into RNA, and RNA is then translated into protein, which actually carries out the metabolic functions of the body. So if you look at the definition of molecular diagnostic, these are nothing but class of diagnostic tests that utilizes these macromolecules like DNA, RNA, and protein for identifying the disease, assessing the predisposition to the disease, monitoring the disease progression, and also for deciding the personalized treatment strategies. Now, in order to understand or comprehend the importance of molecular diagnostic, we can actually look at the global molecular diagnostic market and how that has been growing over the years. So, from the current 16.17 billion,
No.
okay, the next slide. So from the current 16 billions in the current year, it's expected to almost double in the next 10 years, which is 2025. And if you see, the surge is mostly driven by the North America and Europe, as you can see in the yellow and the green bar, which is expected because they are the developed countries doing a lot of work on molecular diagnostic. But if you see the Asian market, which is in gray color, that is also steadily increasing, which shows that the molecular diagnostic is here to stay and is definitely going to grow more better. It's like, so what? Just before that. Yeah. So there are 6 main broad applications of molecular diagnostics. One is the infectious disease. One is the infectious disease. We all know for diagnosis, especially tuberculosis. Molecular diagnostic has reduced the diagnosis of tuberculosis from something like 8 weeks to less than a couple of hours using the specialized molecular assay platforms like the Gene Expert. We then have the oncology where we use tumor-specific genetic alteration to decide the targeted therapy. We Okay, I'm not going to use the pointer anymore. So then we have the thing in the genetic and rare genetic disorders and the lower right, where molecular diagnostic can detect inherited variants, okay, so that we can offer more personalized management in the rare genetic disorders. On the bottom left, we have transplantation as one of the important applications which helps to monitor the donor recipient compatibility. It also helps to determine or the early graft rejection using molecular assays like HLA typing and even cell-free DNA. Reproductive and prenatal medicine, again, molecular diagnostic can detect early the chromosomal and genetic abnormalities and can offer detection using non-invasive testing. And finally, we have precision medicine or personalized medicine, specifically on pharmacogenetics. Now, to be honest, we could talk about each of these applications for hours together, but in the best interest of today's time, I would be focusing my presentation primarily on personalized medicine. So, personalized medicine, precision medicine, pharmacogenomics, stratified medicine, we have come across these terms quite often, but the question is how similar or distinct are they from each other? If you look at the definition, personalized medicine actually utilizes an individual's genetic information to guide therapy, versus precision medicine is a more broader term. It not only utilizes genomic, but it also utilizes environmental and lifestyle information for patient management. But in practice, more oftenly, these two terms are used interchangeably. But precision medicine is something that is used more commonly. And one of the most important domain within precision medicine is pharmacogenetics. It's basically the analysis of an individual's DNA to predict how he or she will respond to that particular therapy. But before we move further, we first need to understand why was there ever a need for pharmacogenomics or precision medicine? And to understand that, let's look at the global pharmaceutical market. There are numerous reports which by different research analysts which keep talking about how the pharmaceutical market is increasing, and it is one of the projections is in the next couple of years the global pharmaceutical market is expected to reach $2 trillion with an annual growth rate of 7%. And this trend is not only seen in the developed countries, but it's also seen the same in the developing countries, including India. But with such rapid increase in the pharmaceutical industry, the most obvious area of concern is the adverse drug reactions. Yeah. If, okay. So as per VG base, okay, this is a W Health organization's database for adverse drug events reporting. On this database, it already has something like 14 million reports from over 180 countries. And if you look at the US FDA, okay, they have a portal, a public dashboard called as the Adverse Event Reporting System, and it's a highly interactive portal. Anybody can log on to this website and can get information about what are the adverse events that are occurring in the United States. And till date, there are around 32 million adverse events which are already reported on the portal on an average of 2 million per year. It's one of the leading causes of mortality in US, and most shockingly is United States spends around $35 billion on the additional medical cost that is associated because of these adverse events. And Europe, on the other hand, spends something like $79 billion. So if such is the grave situation, we also need to understand why do they happen and how can we address them. So one of the major reasons why adverse drug reaction happen is because of the traditional one-size-fit-all approach that we have been using it for years together. In that, what happens is we have a group of patients with the same diagnosis and they have been given the same therapies. By doing so, some of the patient benefits, some has no benefit, while some of them develop adverse event. Now this suggests that there is a large inter-individual variation to drug response. And there are many factors that are involved for this. We could attribute to age, ethnicity, weight, co-concomitant drugs, disease. But the most important factor for this variation is the genetics, the DNA of the individual. The completion of the Human Genome Project in the early 2000s showed that we everybody in this auditorium, we are 99.9% similar in our DNA sequence. But it is the remaining 0.1% that accounts for this large inter-individual variation and also how we metabolize the drug. And these findings has led to a paradigm shift from the traditional one-size-fit approach to a biomarker-driven precision medicine, where the same group of patients are categorized based on the presence or absence of biomarker, which then decides the right dosage and the right drug. And by doing so, we make the treatment more effective, more safer, all patient benefits and the incidence of adverse drug reaction reduces. So how can we address? We've understood why it happens. We need to know how we can address adverse drug reaction. And the answer is pretty simple. We need to identify a biomarker. Now, biomarker, as we know, these are measurable biological indicators. It could be genetic, it could be molecular, it could be any signal that can provide information about the disease presence, progression, or even treatment response. There are mainly 6 types of biomarker: diagnostics, prognostic, predictive, response, monitoring, risk biomarkers. Every biomarker has a different role. But the most important of these from today's presentation is the predictive biomarker, because it lays the foundation for preventing adverse drug reaction, because it identifies patients who are at risk for drug toxicity or non-response, and by enabling dose optimization and safer prescribing. Now, this slide just summarizes how implementing predictive biomarker can transform human healthcare. So, firstly, it takes us away from the one-size-fits-all approach, which we know is inefficient and leads to adverse drug reaction. It supports companion diagnostic, regulatory pharmacogenomic labeling, and that lays the foundation for personalized, predictive, preventive medicine, where the therapy is truly tailored for each individual. Now, we know they are important, but how do we identify them? So this development of biomarker is basically by and large a two-step process. You have the discovery and you have the validation phase. Now in discovery tool, to put it simple, we use high throughput, expensive omics technology to first identify potential predictive biomarker. Once these are identified, they are then validated in the validation phase, okay, in the large clinical cohort, so that we confirm their predictive value. And together, discovery phase and the validation phase, they link the biomarker discovery to clinical validation and its implementation. So what are the tools that are currently available for identifying these? So depending upon the biomolecule that one is interested to identify, whether DNA, RNA, protein, there are different tools that are available. For example, we have on the top left, we have the microarray, which is a chip-based platform, which can allow us to simultaneously detect thousands of genes, thousands of SNPs at a given time. But the problem is, it can only give us the known variants or the known gene information. We then have the next generation sequences. We all must have heard about it, because It is capable of performing whole exome sequencing, whole genome sequencing, RNA sequencing, and all kind of high throughput sequencing. But the biggest advantage of next generation sequencing is that along with the known variants and known genes, it can also identify novel biomarkers. That makes this technology more powerful and more widely preferred. We also have the mass spectrometry, which actually measures molecule based on the mass to charge ratio and allows precise identification and quantification of protein or the metabolites. Now, we have identified the candidate biomarker using any of the tools that fits our interest, and it then moves on to the validation phase. Now, in the validation phase, we use technology which Dr. Anurag had also mentioned about PCRs, digital PCRs. We have the qPCRs. Now, these are techniques which are slightly cost effective compared to the discovery. They are equally sensitive, but they help us to validate the findings from the discovery phase in a larger clinical cohort, because you can't be doing next generation sequencing on a large clinical sample. So you need a technique which is far more specific, far more sensitive, and it's cost effective as well. So these are used. And finally, we have tools such as the DNA sequencer as well as the LC-MS, which confirms these findings and make sure that only the robust, reproducible biomarker moves on to the clinical stage. So the objective is not just identifying them, it's also want to see how it's clinically used. And that's when molecular testing comes into the picture. Now, using molecular testing, we can decide the right dosage and the right drug for each patient so that we minimize adverse drug events. Now, so accordingly, a clinician may have to give, based on the DNA report of that individual, we may have to give a normal dose, and they are called as the normal or extensive metabolizer. In some patients, a lower dose may be required, they are called as the intermediate metabolizers. Higher dose could also be required, which classifies them as ultra-rapid metabolizers. And in some group of the patients, we have to give a totally alternative drug. The other drug should not be given. Hence, molecular testing transforms the pharmacogenetics data into an actionable clinical insight. Now, there's so much of research that has happened globally and it's also ongoing. Where does one actually look for information? Where does the evidence for pharmacogenomics comes from? So we have different, guidelines providers, regulatory bodies. Now they keep curating, standardizing, and updating evidence from the literature for clinical use. And this table summarizes the global ecosystem of pharmacogenomics in clinical use, where the first two, that is the CPIC and the KNMP, which is the Dutch Pharmacogenetic Working Group, they are the guideline providers. They tell us which gene to be studied for which drug metabolism. Then we have the US FDA and the European Medicine Agency. Now these are the regulatory bodies. They directly integrate pharmacogenetic information into drug level. And PharmVar and PharmGKB, they are the drug data repository. Now each of these play a complementary role in curating evidence, defining the allele nomenclature, and also integrating pharmacogenetic information into clinical use. Of this, CPIC is the most widely and internationally recognized for clinical implementation. It was formed way back in 2009. Currently, there are around 550 more gene drug pair which are already listed. And out of these, 139 gene drug pair have strong evidence that one needs to study those genes before the drugs are being tested. They are peer-reviewed, freely available, and they are constantly updated regularly. So there are many guidelines which are already being published. For example, for warfarin dosing, you need to study for CYP2C9 and VPORC genes. For tacrolimus phenotype for dosing, you have to study the CYP385 genes. For clopidogrel, which is an antiplatelet drug, you have to first study CYP2C19 gene. And so on their website, there are something like 29 already published guidelines. We then have the US FDA. Now, the US FDA provides the strongest validation of pharmacogenomics in the clinical use. Currently, there are more than 600 FDA-approved drug labels which already contain information for pharmacogenetic biomarker, of which majority of them from oncology, urology, infectious psychiatry, and so on. And if you see the distribution of this 600, those in the red box, which accounts to something like 146 drugs, for them, there are direct actionable implication for either you give the medicine or you don't give the medicine. These are just some of the few FDA-approved biomarker-driven targeted therapies options. Now, each of these drugs are designed for a specific patient harboring a specific genetic mutation. If that genetic biomarker is absent, this therapy will not work. So now comes the problem. We have the problem of plenty. So we have the guideline from CPIC, we have guidelines from AFP, Dutch, PharmGKB, so many things. But that will be so cumbersome, so irritating that you have to go onto these websites, look for the drugs that you are interested, interpret that and use for clinical use. So would it not be useful to have a portal which has everything in one place? And that's where ClinPGX plays a very important role. It is an integrated, NIH-supported resource platform that consolidates the pharmacogenomic evidence from guidelines, drug labels, and it helps to take decision-making clinically. So there are many tools which are already available on the portal. The first one is, I'll just name a few of these, which are called as the PharmDog. That stands for Drug Optimization Guidance Tool. This tool allows us to search in terms of genetic test or the drug name. So for example, if we enter the drug name azathioprine, which is anti-inflammatory, it tells us what are the genes that one needs to study. And once you enter the result of that gene, it actually gives you information for that particular drug gene in terms of what are the CPIC guideline tells you, what is the Dutch guidelines telling you, and what does the FDA label tells you. Then you have another tool. Okay. Then you have another tool called the Genotype Selection Interface. Now here you enter the gene information first, and then when you click on Make Report, it tells you what is the metabolizer status. It tells you which are the drugs for which these genes are responsible. And the best part is this. It actually gives you a comparative table from all the different resources and gives you a compilation of all the guidelines. I think this is one of the best tools that they have, because in one snapshot, you could tell what does CPIC mentions, what does Dutch guidelines says, what are the FDA labels, and what are the other FDA guidelines. The other one, this one is more like a bioinformatics. Those who do a lot of bioinformatics and next generation sequencing, you just have to upload the raw data onto this portal, and it can tell you what are the alleles, what are the functions, what are the phenotypes, and it can also tell you for that particular patient what is recommended as per the guidelines. Now we have all the resources, we have all the guidelines for its effective implementation. So let's look at some of the studies which have been carried out globally. So there are several large-scale studies that have been conducted where they have successfully shown implementation of pharmacogenomics in clinical care. But if you can see, most of these are centered in the United States or in the Europe. Representation from the remaining part of the world, like the Latin America, Africa, Asia, and Middle East, is very limited. And why is that so? Why is it limited? Because there are many barriers to its effective implementation. One of the key barriers is the diverse population, the underrepresentation of the sample. These guidelines are yet to be integrated into the clinical workflow. There are issues with data integrations, and I think one of the primary hurdles that I personally have come across is the acceptance by the clinicians for using these kind of support of pharmacogenomic data. And I'll just quickly take you through a few of those studies which talks about these barriers. So there was a survey that was conducted on pediatricians from US and Japan, and shockingly, only 10% were familiar with the concept of pharmacogenetics, and only 10% knew about what these CPIC guidelines are. But on a positive side, most of them believe that pharmacogenetics would improve efficacy, would improve safety, and would of course be interested in learning more. Another survey, a systematic review was conducted on 52 studies from 26 countries or 12,000 plus pharmacy students. And more than 65% of the studies, the participant responded their own knowledge as low or insufficient. And more than 70% requested more structured pharmacogenetic information in their curriculum, and that should be the way forward. Another barrier was the underrepresentation. So in this, the authors extracted the allele frequency data from 23 CPEC guidelines, which include 19 genes, and they compared the representation across 9 biogeographic groups. So here, if you could see, the genes are listed on the x-axis, and the populations are listed on the y-axis. As you can see, for most of the populations, whether it's the Latin America, the Southeast Asia, or the Oceania, they are quite underrepresented in these guidelines, which means these guidelines have been made using samples that are not primarily from all over the world. They are predominantly based in Europe. So as you can see that, out of these 19 guidelines, 16 guidelines were from the European population, and most of these populations were quite underrepresented. So, because these guidelines do not represent a population, it becomes very important that we identify and validate population-specific biomarkers in the clinical relevance. And why is it important? Because validating one biomarker in a given population might not accurately predict its outcome in another population, because that will lead to misclassification, and we need to make sure that the precision medicine is more personalized and it's for all the well-represented studies. It improves the predictive accuracy, enhance clinical safety, and support regulatory acceptance. In order to bridge this global genomic diversity gap, in recent years there have been quite a few studies. Like we have the account trial, which was based on the African American inflation. Then we have the UAE Arab Pan Genome Reference. We have a large study on a large Chinese cohort. And then we have a very own Genome India study. Thank you. And then we have a very own Genome India project, which was published early this year. And the accounts trial happens to be the first PGX implementation study focused on the underrepresentation population. The second, which is the Taiwan Precision Medicine Initiative, one of the highlights was in this 46,000 individuals, 99.9% carried at least one actionable variant. And if you look at the last Genome India project, it gave us something like 140 million variants. That is huge. Okay, so this was the first time that these kind of nationwide genomic studies gave us the population-specific DNA variants which were previously missing. And now, to further emphasize the importance of validating predictive biomarker, I would like to discuss our own study that was conducted in the hospital. This study was conducted on thiopurine drugs, which is one of the most commonly used anti-inflammatory drugs. And one of the problems of this drug, it has a narrow therapeutic index. And there is an enzyme called as TPMT, that's thiopurine methyltransferase, which is very important for the drug metabolism because it maintains a balance between the active and the inactive component of the drug. Now, this enzyme activity, which is play an important role in the drug metabolism, it shows a variation which leads to increased drug activation and severe toxicity. And this variation is attributed to the presence of DNA mutations in the TPMP genes. And that's why in 2011, CPIC and the US FDA strongly recommended TPMT genetic testing prior to thiopurine dosing. So we started doing it in Hinduja Hospital since 2009, I would say. But our experience was that majority of the patients stopped this thiopurine drugs due to side effects, primarily leukopenia. We found that patients with TPMT mutation accounts for only 33% of the leukopenia. So all the leukopenia that we have seen, but because of thiopurine, only 33% of the patients had TPMT mutation. The remaining 67% had no TPMT mutation, which means that the TPMT frequency is lower in Asia compared to the European population, but the incidence of leukopenia is much higher. And this makes us think that how useful is TPMT testing, especially in patients of Asian origin. We recently published our finding of almost 1,014-year experience from a tertiary hospital in the European Journal of IFCC. So our question was, if TPMT is not useful, could there be any other additional genetic factor that actually be more better? And in 2014, there was a microarray based study on something like a 978 Korean subjects with Crohn's disease. And what they found was something very, very unique. They found a gene variant in NUDT15 which was extremely significant with the risk of developing thiopurine-induced leukopenia. So we thought, if the Koreans had found it, is it possible that we may still have that in our population? Is it more common in the Asians? So with that in mind, we conducted a study to determine the association of this variant in our population in Indian patients and to see whether DPFT or NUDT or both can be used to better predict the toxicity in Indian patients. So it was a retrospective study. We already had the DNA samples for patients who were coming for TPMT testing. We performed a simple PCR-based assay. We validated by DNA sequencing. And this was a small cohort to begin with of 111. These were the clinical conditions and the mean thiopurine dose. But in this cohort, there were 7 patients who developed leukopenia, of which 5 were within the first 8 weeks and 2 were after the first 8 weeks. 4 of these developed alopecia, of which 2 had severe hair loss. These were the adverse side effects that were seen in this cohort. We saw in this cohort, when we looked at TPMT mutation, none of these cohort patients had the presence of TPMT variant, which substantiates our observation that the variant frequency is low and TPMT might not be clinically relevant in our population. But if you look at the NUDT15 allele distribution, almost 10% of this cohort had present this NUDT15 variant. We then wanted to see how is the association of leukopenia with the variant. So of these 7 patients who developed leukopenia, 5 of them were heterozygous, which means they had one normal allele and one abnormal allele, and 2 of them were homozygous mutant, which means they had both the alleles as mutated. Now, if you look at those left-hand side, those who develop leukopenia and were heterozygous, they developed leukopenia within the first 10 weeks, but they recovered from leukopenia when the dose was reduced to half, and they eventually responded to the therapy. On the other side, those who were homozygous mutant, which means they had both the copies mutated, they developed pancytopenia, early severe loss within the first 2 weeks, and they recovered from these side effects upon the stoppage or cessation of this drug. So in these 2 patients, the drug had to be stopped, otherwise it was extremely life-threatening in these patients. We looked at it statistically, it was extremely significant with the risk of developing toxicity. And we published a finding in 2017, happens to be the first from the country to find that association. In 2018, we did another larger study because the first study was a smaller cohort study, and we found the same association. And we, in our hospital, started preemptive NUDT15 genotyping into the routine clinical care. And the dose was reduced to half in patients who were heterozygous. Now, around that same time, there were numerous other studies from Asia, like from South Korea, Japan, Taiwan, you have from Thailand, Lebanon over there, but the Lebanon allele frequency is extremely low, from China and India. Now, with so many studies coming from Asia, such was the impact that the same guideline which earlier said only do TPMP, they were compelled to update their guidelines including TPMP and NPP genotyping. Now at Hinduja Hospital in Mumbai, we are constantly doing research on pharmacogenomics, and those which are actually clinically relevant directly comes into the routine molecular diagnostic for patient care. So right now, these on the top you can see the drugs for which there are different genes that are involved, and we are offering this as diagnostic services to our patients. So let's look at what the future direction in precision medicine would be. Now, in order to derive holistic predictive biomarker and personalized therapeutic insight, it's high time that we have to perform a multi-omics integration, wherein we combine the data from the DNA, the RNA, the protein, the metabolites, so that we understand the comprehensive biological signature, and we can help to stratify patients into molecular distinct subgroups and enhance the predictive model for drug efficacy, toxicity, and disease progression. But having said that, integrating such vast omics data can be very, very complex and very difficult. It's beyond the existing statistical modeling which are already available. And that's where we would have to depend on machine learning and artificial intelligence, because they can integrate and convert the raw genomic data into clinical and actionable predictive model. They can better predict the adverse drug reaction, enhance the clinical decision support, and accelerate the biomarker discovery process. So, machine learning has the potential to transform the pharmacogenomics from reactive interpretation to predictive intelligence, which is actually the true foundation of next generation precision medicine. So my final slide, the final take home from my presentation is the molecular diagnostic is definitely the foundation of precision medicine. But precision medicine will only succeed if this molecular information is translated from the bench to the bedside. We have pharmacogenomics to help us address the issue of adverse drug reaction. We have different guidelines which are available, but we need to validate those guidelines in our population, because Indian population needs India-specific population variants. We need to overcome many barriers in terms of awareness, acceptance, technology, integration, and by doing so, hopefully, in the near future, we will have successful PGX implementation, and that can lead to reduced adverse drug events. I would just like to end my talk with a quote from Sir William Osler. He says that it is much more important to know what sort of a patient has a disease than what sort of a disease a patient has. And this is so apt. So I would like to thank you all for your time, your attention, and if there are any questions, I would be happy to take. Thank you.
I must congratulate you for such a brilliant talk.
Thank you so much, sir.
You have given the whole idea of precision medicine in a simple words. You have dealt with predictive biomarker, which is the future of our medical science. And rightly you have covered that a lot of data is there, and that has to be analyzed by machine learning and AI.
Yes.
So there is a concept,
Yes.
predictive indicator, you have the biomarker.
Yes.
But nowadays, within 5 years, I think there will be predictive medicine.
Absolutely.
It is only precision medicine, targeted therapy, tyrosine kinase inhibitors, and the
Sure.
So you can give uh, precision medicine, absolutely. But there are certain areas where you require predictive medicine. For example, we have already said about CYP metabolism.
Yes, yes.
For undergraduate students, you are saying the slow acetylator of INH
Yes.
and fast acetylator of INH, Japan and India. So that is required. Yes, also we should have a genomic bank. Each and every person's genomic profile.
Yes.
How much it costs? First, my
Yeah.
question is,
Yes.
how much it costs
Yeah.
and where is the Indian any firm or that can give me the data genomic profile? For example, मैंने अपना कराया,
Yes.
मेरा मालूम हो गया, CYP क्या है?
Yes.
या even HLA है.
Yes.
You can predict. There are many drugs which can cause, as you have already told about myopia. There are many drugs which, for example, comorbidity. So these are the, you can prevent such type of adverse reactions. So how we should pursue? For example, हमको लेना है comorbidity.
Yes.
हम कहाँ जाएंगे? या हम IRS ले रहे हैं. हम कहाँ जाएंगे?
Yes.
Yes. And that then only आप बहुत अच्छा कर सकते हो.
Yes.
मतलब एक, that's why I have given you have touched very sensitive topic and future of the medical science.
Absolutely.
Thank you.
So I need to thank you first because this is an excellent question and I was hoping somebody can ask me this question. The more simple it is to answer this question, the more difficult it is to address this question. So your question about there are so many drugs, anybody can get himself or herself tested and then do what? And you also have a question on the cost. So number one, in terms of cost, just to give you a perspective, the Human Genome Project, which was completed took 30 years to complete, they spent something like $3 billion just to sequence one individual's DNA. Now the same cost has come down to less than 12,000. Yes. So that's the whole exome sequencing. So if the whole exome sequencing costs something like 12,000, what we are interested in the predictive biomarker, which could be a very small percent of the whole exome. Right? So my personal understanding and my personal experience says that the cost for testing an individual for all the known, published, validated predictive biomarker should not be more than 10,000. Yes. So that's one thing. Now, the same concept, what you asked, is already being implemented in a lot of these European countries. Okay, especially I happen to know one of the collaborator, Professor Ron Schack. He's from Erasmus University in Rotterdam, Netherlands. And almost a decade ago, he did a pilot study on a group of individuals and they screened the known variants in that population and they generated a DNA passport. That's what he called it. And that DNA passport information is relied to all the pharmacy stores in that particular city. So if a given individual happens to be given by the physician a particular drug that, for example, you need to be given as a therapeutic, he and she, as and when they went to the pharmacy store to buy the medicine, the pharmacist will have the information on his or her portal saying that this individual is a poor metabolizer, a novel metabolizer, or an intermediate metabolizer. And along with that, the guidelines stating what should be the dosage based on the metabolizer state already on his system. So, if an individual walks in with that DNA report, the pharmacist should be able to tell him, yes, you need half the dose. You don't need a 50 milligram dose, you need a 25 milligram per dose. And accordingly, the therapy is given to that particular patient. Now, this was done in a very small pilot study, but it's very effective. Now we are talking about our nation, a 1 billion plus population. Now, this is easily possible because we recently completed the Genome India initiative on 10,000 individuals. So, we should be able to identify which are the most common pharmacogenetic predictive biomarkers that are seen in our population, right? The variants could be thousands. We need not sequence thousands. We need to sequence only those which are seen in our population. So, it could be something like 50. You need to sequence every individual for that 50 variants, and maybe at the moment that individual can keep that information with himself before it becomes more nationwide and integrated into our electronic medical record system. So there is much more to talk about this, but it's going to be a challenge. But in the recent years, there are a lot of these initiatives which have come, and those are very promising. So I'm very hopeful that in the next decade or so, we should be able to sequence in a most cost-effective manner all of us so that we can prevent and reduce the cost that we spend on these adverse drug reaction patient management. So yeah, I hope I've answered your questions as possible.
So for a single gene study,
Yes.
which technique would you suggest? Is it cyber sequencing or PCR or next generation sequencing platform?
So it all depends. So here the selection is based on the technology and the cost. See, no matter what you do, the cost will come into a picture. So if your gene, do you want to study the entire gene, right? So if the gene is like 25,000 bases, a standard sequencing will take a very long time because standard sequence can only sequence 500 to 600 length at a time, right? And if you're wanting to sequence a 25,000 nucleotide long, standard will not be the most appropriate one. Then, and nowadays, to be very honest, nobody actually studied one gene. Because of the potential and the power of next generation sequences, you can actually study 25 to 20,000 all the human genes in less than 12,000. So why would one spend 6,000 studying just one gene versus 12,000 studying all the 20,000 genes? So, but again, if you're studying a gene for studying a specific variant, a DNA mutation, then definitely a PCR-based method would be the best thing. Then you don't go for NCS. So if you know, for example, the one that I showed about NUDP15 and PP15, we do a normal PCR gel-based methods for our testing because that comes out to be very cost-effective. You may have all the tools at your disposal, but you have to see for patient management, the cost is very important. You cannot develop something for 20,000 and expect the patient to pay that much. So the more cost effective it is, the more easily accepted and widely used it will be. So I think it all depends on what are you studying and whom are you catering to. If you're doing just for research, fine. But if you're looking for patient management, you have to keep in mind the cost that is associated with it.
A single gene variant using PCR.
Average single cost, somewhere like 2,000 to 3,000. The one that we do for NUDP15 in our hospital is 2,600. That's a single variant that we study. It requires, yes. So that's a one-time cost, sir. That's a one-time. But I think I could not show one aspect because of lack of time, the POCT concept. So there are these small point of care testing which are commercially available that could be kept in any clinics or any mobile health ambulance. You just put the patient blood sample and it does the extraction and the genetic testing in that small machine only for that particular variant and will release the report to you. So there are already FDA approved. In fact, in my slides, there will be one slide. So POCT, if you go, will be expensive. POCT would be expensive. So POCT establishment is not a cost invasive process because it's just an instrument. You can just place it here and. But in India, to be very honest, I have the prices for Europe because I've been attending. So they charge anywhere between 80 to 90 euros for one report. But in India, The POCT for pharmacogenetic testing is not that widely used. Everyone prefers to establish a basic laboratory, right? Invest for one time, and then you can make all your investigation cost effective. But yeah, it depends. Sorry, I didn't hear you.
This medical college want to establish that. What is the cost? I want that. Can you popularize patient, pay for it is essential for patient safety.
Yeah.
That's what.
But my personal advice for an institute like this, of high capacity and bigger aims and objective, POCT is not something that an institute of this caliber should go for. You should actually look for establishing a molecular lab. Yes, and because POCT will restrict, yes, just one or two, right? Versus a one-time established lab can do any number of things under one roof, right? That would be like a center of excellence. Versus POCT will only be do for one pharmacogenetic test, two, three, four, that's it. But we have hundreds of tests to be done, and pharmacogenetic is just one of the applications. As I mentioned, infectious disease, oncology. I think the imagination is only a limitation. The bigger we can think, the better we can serve. So
So one more question.
yes, who's that?
So thank you for insightful presentation. It was very informative. I want to ask that what's the implication of pharmacogenetics in India in detecting the cancer presently? What's the present status?
So, as I said, if you see the US FDA slide which I showed, the US FDA is already, currently there are more than 600 drugs for which there is pharmacogenetic information already available, of which almost 35% of the drugs are related to oncology. So, and there will be some examples like cetuximab, panitumumab. There are so many targeted therapies which are only useful and effective in patients who harbors that specific DNA mutation. If that DNA mutation is absent, the therapy will not work. So definitely it has a lot of scope in India.
So right now, our clinicians and the doctors are using it on patients that, like, if breast cancer patients are
Absolutely.
so
Absolutely.
it is
Yeah.
a-
So for example, you talk about breast cancer, you have the BRCA1, BRCA2, and
Yeah.
all the other molecular tests. And based on that, the presence or absence of these BRCA gene mutations, there are definite specific targeted therapies. Okay. One patient has BRCA1 positive, you need to give. Second one does not have, you don't give that therapy. You start the conventional chemotherapy and the surgical options you may have. But these,
So is it cost effective right now?
it varies from place to place, lab to lab. But as I said, whole exome sequencing, which if you know the genomics, you have the whole genome. 2% of the whole genome is exome, and even 0.1 or 1% of that would be the targeted panel for oncology. So you have for oncology, for all the other types of lung cancers and breast cancers and colon cancers, colorectal cancers. So depending what would one actually want to serve, so but the cost can vary. It could be anywhere between.
So if it is getting implicated in today's time, then why the cases are getting so high?
Because not everybody has the same mutation.
Yeah, so we are doing now that personalized medicine is if it is getting implemented, then why the cases are rising so high?
But it is a growing field. It's not fully developed. Not every laboratories are doing.
Yeah, that.
Exactly.
Yeah.
That is the barriers which I talked about.
Yeah.
There is the barriers. There are problem with, I would say, the acceptance. Even in my own hospital.
Yeah, that I
In my own hospital, I will share my own experience. In my own hospital, I am very thankful to a gastroenterologist who, for their patients on inflammatory bowel disorders, they do the CPMT and UDP preemptively. before starting the therapy. But I also have the cardiologist who refuse to accept that there is CYP2C19 genetic test to be tested before giving clopidogrel. But at the same time, I have a neurology physicians who are doing it for their patients on stroke. So it all depends about how you convince them, how you.
So why is it not getting universally accepted?
There has to be more nationwide involvement. I personally feel that is where I feel there has to be something from the ministry to lay down certain directives, guidelines that yes, one patients to be given a particular drug, these genetic testing is very important. There has to be some medical legal implications for not doing it. But because these practices are very different in different parts of the country, in different states, different cities, you're going to see this very fluctuating workflows in patient management. So you talk about the tier one city in Mumbai, in Delhi, and in the other top cities, you would see these technologies being adopted and the clinicians are more proactive. But you go in the second tier, the third tier, it is the lack of awareness, the willingness, the cost that actually are those obstacles for its effective implementation. So there are many challenges. So the silver lining is we are getting better day by day. So maybe in the next decade or so, we will be in a far better position and far more equipped to address and implement precision medicine.
Thank you so much.
Yes, thank you.
Thank you very much, sir, for sharing your vast knowledge and expertise with us today. We are truly grateful for your words and the valuable perspective you shared with us today.
So with this excellent session, we conclude our CME talk. We're truly glad to have all the three esteemed speakers with us for today's session. We are honored by your presence as we begin with this significant occasion of inauguration ceremony. We will now break for the tea and prepare for the upcoming ceremony. Please join the break and we look forward to convening shortly.
इसके बाद क्या इसके बाद क्या रहेगा इसमें कैसे देखो सब हमको मिल गया नहीं पूरा मिला के 40 पूरा मिला के 40 रुपये हमको मिल जाएगी वो पावर हाँ
लाइटिंग का लैम्प होगा ना तो गाना बजा दीजिएगा।
वो पेन ड्राइव मांगता
सर को पैसा दे दिया है।
कहाँ लगा सदा गोम की बुलेट गोम ये बुलेट गोम ठीक चेक कर
भाई इट्स हेल्प
पहले चेक करवाइए, चेक करवा के उसको लिए करवाइए। लेके जाके
उसको अभी न्यू पैनल में चलाना है, पीपीटी में भेज दो, पीपीटी कर रहा है, पावर पॉइंट ये कितने बजे तक ये खोल देंगे। ये पैनल डिस्कशन में चलेगा। अब लोगों डाउनलोड हो गया लंच के बाद चला वो एक बार होने वाला है मेरा डाउन करो पूरा पेज नहीं है कौन सा फील हुआ रहता है थोड़ी फटेंगे
ठीक है, 40 में चालीस मतलब आपका कोशिश करना चाहता आदमी के लिए जो रेट है 40 सॉल्यूशन सॉल्यूशन बनेगा 25 करने का मतलब जैसे दो आपको ठीक है इंस्टॉलेशन ठीक है समझ गए और 25 कैमरा का है तो 25 कैमरा को मैंने प्रेजेंट कर दिया ठीक है एक तो प्लेसमेंट का होता है ये सिर्फ ये बता रहा है baseball. Once we do it, within 194. I got the clip. Making लंबी बोली. से और बेटर कर सकते हैं और क्वालिटी अच्छी चला पा रहे हैं ट्रैक वीडियो वो भी आ रहा है हमको फायदा हो गया हमको बहुत आ गया किसी का व्हाट्सएप नंबर मुझे दीजिए ना, बता देते हैं उसी को करेंगे। किसी का व्हाट्सएप नंबर दीजिए है ना, अभी अगर
नहीं नहीं, हम मेडिकल कॉलेज बिहटा से बोल रहे हैं। पहले ये कंफ्यूज दूर कीजिए, ये पटना में कॉलेज है। जब देखिए आप नेताजी सुभाष मेडिकल कॉलेज बिहटा, हम ये
भेज दीजिए। आपको बोलिए फोन करने के लिए।
हाँ, जो खाली सब बता दीजिए, जो खाली है
ठीक है। तो उससे हाँ हाँ, हाँ 138 प्लस 40 ये पेमेंट होगा आपका, ठीक है। हाँ हाँ, क्या नहीं भाई, इसी में 138 में ही आपका वो उसी से पहले ले लिए थे। हाँ 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 हाँ, उसी में उसी में 153 का इंस्टॉलेशन रिपोर्ट बनेगा, जो 600 रुपये के हिसाब से वो अलग से नहीं है, उसी में हम आपका ले लिए थे ना, उसके बाद वो आके लगाए थे। और चेक करा लेते हैं, चेक करा
इसको हटा दीजिए और जो
Attention please, we are going to start the inauguration ceremony. All of you are requested to kindly take your seats.
हाँ, और अभी तो जो
बता देते हैं हो जाएगा ना हो जाएगा ना
ठीक है तो फोन पूछ रहे
दोनों चालू जो फालतू दो ही बचेगा 10 सवाल
पुनर्मित्रं पुनर्मित्रं पुनर्भाया पुनर्बहि एतत् सर्वं पुनर्लभ्यं न शरीरं पुनः पुनः धन संपत्ति मित्रगण पत्नी तथा जमीन जायदाद की हानि होने पर यह स्पीड जा सकते हैं, परंतु परमेश्वर द्वारा मानव शरीर एक बार नष्ट हो जाने पर पुनः उपलब्ध नहीं हो सकता।
In the grand tapestry of human existence, many essential elements, fortunes, vital relationships, and profound love can often be successfully repaired and restored. Yet a single critical filament exists, which once fully severed is beyond measure. That is our physical health. The human body is indeed a complex and extraordinary mechanism capable of remarkable feats. Nevertheless, its inherent fragility makes it highly susceptible to deterioration. Therefore, to unlock and sustain both peak performance and enduring longevity, the dedicated investment in its absolute well-being remains an unwavering necessity.
This is the precise motivation for our gathering here today. This distinguished summit underscores our unwavering dedication to push the boundaries of health and wellness. Together, we are poised to explore the latest research, disseminate truly groundbreaking ideas, and establish the strategic partnerships that will catalyze the future process of diagnostics. By strategically uniting our diverse expertise and resources, we hold the potential to profoundly revolutionize healthcare and enhance the well-being of individuals across the globe.
Honorable dignitaries, esteemed guests, respected faculty members, and fellow delegates, it is our utmost honor to welcome you all to the Bihar Molecular Diagnostics Excellence Summit 2025.
This marvelous symposium convened on December 12th with intensive workshops on digital PCR and NIPT, along with immunohistochemistry and breast cancer. Powering through the 13th with various scientific sessions by our marvelous guest speakers from around the country. All leading up to this grand inauguration ceremony, where we have the brilliant opportunity to be blessed by such myriad of eminent personalities.
The theme of this year's summit is From Markers to Molecules, capturing the scientific evolution that defines the modern era of diagnostics. For decades, biochemical markers served as the foundation of clinical investigations, providing essential clues that guided diagnosis and management. However, as our understanding of disease has deepened, the limitation of conventional markers have prompted a natural progression towards more refined molecular level insights. Today, diagnostics allows us to look beyond what is visible at the biochemical level and explore the very architecture of disease.
The theme is therefore not just a scientific statement. It is a reflection of progress, precision, and the expanding capabilities of modern laboratory medicine. It signifies our commitment to moving from broader indicators to deeper molecular understanding, ensuring that healthcare in Bihar and beyond becomes more accurate, evidence-driven, and patient-centered.
And catering to this idea is the host of this marvelous gathering, prestigious institute of Netaji Subhash Medical College and Hospital. Established by Sirpanto Devi Mahila Kalyan Sansthan and Amara Bihta, this institute is the brainchild of our visionary chairman, Shri M.M. Singh sir. Their idea behind establishment being affordable, best-in-class medical education to the students and nurture world-class doctors for the service of the nation. Thus, giving us the philosophy of our college, Seva Paramo Dharma.
And this gives us the perfect opportunity to recognize individuals that have devoted their entire life to selfless service. In the tapestry of life, there are threads woven with exceptional dedication and unwavering purpose. Tonight, we gather to honor those whose lives have been such threads, enriching our community with their tireless dedication and unwavering commitment.
With great honor and anticipation, we invite the distinguished guest, Dr. Anurag Kumar sir, Assistant Professor, Biochemistry, AIIMS Patna, to grace our event.
With great pleasure and respect, we now extend our invitation to our distinguished guest, Dr. Swaroop A.V. Shah, sir, Senior Senior Molecular Scientist, PT Hinduja Hospital and Medical Research Center, Mumbai, to join us on the dais and grace this occasion.
We now welcome the Honorable Managing Director of our institute, Shri Krishna Murali sir, the man who has flown to stars but still remains well rooted to the ground. Please join us on the dais.
It is our honor to invite the principal and backbone of our institute, Professor Dr. Harihar Dixit sir, to grace the dais with his presence. I now welcome the Dean and Head of Department of Radiology, our institute, Professor Dr. Prem Kumar sir, to join the dais. Sir is an indispensable element in the functioning of our academics.
It is our honor to invite the organizing chairperson and a cornerstone to this summit, Head of Department of Pathology of our institute, Professor Dr. Rajesh Kumar sir, to grace the dais with his presence. I am now privileged to invite on the dais the enigmatic and visionary organizing secretary of this grand summit, Subdean Academics and Research, Lab Director and Associate Professor Biochemistry of our institute, Dr. Srinivas Singh.
We are privileged to have amongst us our distinguished guest, Dr. Anurag Kumar sir, who is the Assistant Professor of Biochemistry at AIIMS Patna. Please accept this bouquet, sir, as a token of our respect and admiration. I humbly respect our Managing Director, Shri Krishna Murali sir, to present Dr. Anurag Kumar sir with a bouquet. It is our honor to invite the medical superintendent of our college, Dr. Uday Narayan Singh sir, to please grace the dais with his presence.
We also have the privilege to have amongst us eminent Dr. Swaroop A.V. sir, Senior Molecular Scientist at Hinduja National Hospital and Medical Research Center, Mumbai. I request our respected Managing Director, Shri Krishna Murali sir, to present sir with the bouquet. Please accept this bouquet, sir, as a small gesture to express a heartfelt appreciation for your presence.
We express our utmost respect to Honorable Managing Director of our institute, Shri Krishna Murari sir. I request our principal, Dr. Harihar Dixit sir, to please offer him a bouquet as a token of her heartfelt appreciation for his presence.
As we embark on this intellectual journey together, let us seek the blessing of the divine to guide our endeavors. I invite our esteemed dignitaries to join ahead in lighting of the lamp of knowledge, symbolizing the illumination of minds and pursuit of wisdom. This ancient tradition of lighting of the lamp signifies the dispelling of darkness and the dawn of enlightenment. May the flame of this lamp inspire us to explore new frontiers of knowledge. Enriched by the blessings of the Almighty, with divinity surging within us, we would like to take this opportunity to call up to the podium the organizing secretary of this summit, Saudi Academics and Research, Lab Director and Associate Professor Biochemistry of our institute, the timeless, ever gracious Dr. Swanima Sikna.
Very good afternoon, everyone. On behalf of NSMCH, it is my privilege to welcome our esteemed dignitaries on the dais, our distinguished guest speakers, MD Sir, Principal Sir, Dean Sir, MS Sir, our organizing chairperson, and I also extend a warm welcome to all the HODs, faculties, residents, postgraduate students, interns, and all the MBBS students present in the hall. The Bihar Molecular Diagnostic Excellence Summit 2025, Chapter 1, marks an important academic step for our institution and for the state. Yesterday's hands-on workshop on digital PCR and immunohistochemistry, we had expert trainers from Chennai and Hyderabad, and saw exceptional participation and enthusiasm from the participants. This CME and workshop is exemplary because it brings cutting-edge diagnostic technologies directly to Bihar, so that creating a platform with these advanced technologies to the real clinical scenario where it will be utilized. Such academic initiatives are invaluable in helping the doctors and students stay aligned with the rapidly evolving field of precision medicine. Today, we are honored to have distinguished guest speakers, Professor Dr. Arnab Pal from PGI Chandigarh, Dr. Anurag Kumar from AIMS Patna, and Dr. A.V. Swaroop from PD Hinduja Hospital, Mumbai, whose expertise has greatly enriched this summit. On behalf of the organizing team, I sincerely thank MD Sir, who has left no stone unturned and is always there to be fulfilling all our requirements regarding this CME and workshop. And with this, I again welcome you all to this summit. Thank you very much and have a good day.
Thank you, ma'am. We express our sincere gratitude for your elegant address. It is a profound privilege to present and invite the organizing chairperson of this grand summit, SOT Pathology, Professor Dr. Rajendra Kumar sir, to welcome the audience.
Good afternoon, everyone. I will from my side the faculties from the those who have presented. Dr. Arnab Paul from PGI Chandigarh, Anurag Kumar from AIMS Patna, and Dr. Saab from India Hospital Bombay. From my side and from our department side, I welcome you all and thank you. Thank you.
It is a profound honor and great anticipation that I invite to the podium the Dean and Head of Department Radiology of our institute, Professor Dr. Prem Kumar. Sir is the epitome of disciplined rigor and indispensable element in the functioning of our academies. Please join us.
On the dais, off the dais, and delegates of this conference, our guest speakers, my thank you for this gathering, and I invite everyone to join this CME and conference on every time this CME is more prominent for with the advancement in medicine, the molecular biology has significance, immense significance, and as the humans are largely being understood for the infectious diseases, now it is time for the molecular biology to take over this. So I congratulate everyone for this conference, in this conference. Thank you.
It is our singular and profound honor to invite the principal of our institute, Professor Dr. Harihar Dixit sir, to the podium and please give the welcome address.
Thank you. Respected Managing Director, Mr. Murari sir, guest Dr. Shah, Swaroop A.V. Shah, not Shah, from Hinduja Hospital, Dr. Anurag Kumar, Dean, Dr. Prem, Chairman of Managing Committee, Dr. Rajendra Rao, Medical Superintendent, Dr. Udayan Singh, and the Organizing Secretary, Shorty. Good afternoon to all of you. I must congratulate the organizing committee for organizing such a beautiful CME on molecular diagnostics, and they have chosen a very updated topic which is very close to me, that is integrating molecular diagnostic into precision medicine from bench to bedside. The future of medical education is not only where we are thinking about the precision medicine, but as my view, it is predictive medicine. Because future will be on predictive. How you can predict the diseases which are one of the important factors of hospitalization, not only the drug diseases, they are, of course, they are cancer, metabolic diseases, but the important third or fourth reason is drugs in USA or even in India. 10% IPD due to the adverse drug reaction. 10% IPD, it is a, क्योंकि हमारे यहाँ का डाटा अभी मिलता नहीं है, बहुत लिमिटेड डाटा है और उस पे हम लोग काम करते हैं, तो you can predict adverse drug reaction. You can predict diseases also. HLA, DQ can predict severe diseases. Also, you can predict ankylosing spondylitis. These are the diseases. And you can prevent, as I said in interaction with you, Dr. You can prevent the adverse effect of carbon. If you have a HLA type, if the patient is having HLA B1502, then you can, ये मालूम हो गया कि ये HLA B1502 का है patient, तो काला में नहीं देना, because it can lead to the event of syndrome. Similarly, HLA typing you can prevent the adverse drug reaction of hepatitis. So there will be a, I think, within 5 years, taking the genomic data of individual and analyzing by machine learning, that is artificial intelligence, you can save the patient. आर्टिफिशियल इंटेलिजेंस अभी गवर्नमेंट ऑफ इंडिया ने आपका और ये सब कुछ किया है और जीनोमिक बैंक के साथ जोड़ा जा रहा है। एज यू हैव सेड ये 12,000 के आसपास आएगा, लेकिन रिस्ट्रिक्टेड होगा, लेकिन ये डाटा हमारे यहाँ है अवेलेबल, हम अपना कराते नहीं हैं। उनसे जैसा मुझे जानकारी है कि 1.5 लाख के अंदर में हमारा जीनोमिक प्रोफाइल बन सकता है। अगर ये हर आदमी का जीनोमिक प्रोफाइल हो जाएगा तो वो हम बीमारियां रोक सकते हैं जो ड्रग के कारण होगा, या एंटिसिपेट कर सकते हैं कि ये बीमारी हो सकती है। It is very close to me, this topic, because I am a founder president of STIGMS, the Academic Society of Pharmacologists. We are organizing conference in November at Varanasi BHU, and one of the important sites is here at Netaji Subhash Medical College also. उसका टॉपिक है Beyond Alert, Predictive Intelligence for Rational Pharmacotherapy. Theme is Beyond Alert, Predictive Intelligence for Rational Pharmacotherapy. एक वाइस चांसलर के साथ हम लोगों की मीटिंग हो रही थी, तो जो मैंने आपसे पूछा था कि कॉस्ट क्या होगा, बिहार में मॉलिकुलर इंदिरा गांधी इंस्टीट्यूट ऑफ मेडिकल साइंसेज में देखा है, यहाँ माइक्रोबायोलॉजी के अंदर में एमआरयू लैब चल रहा है। यदि पीएचडी माइक्रो आरएनए पर काम की है और वहीं से की है, माइक्रो आरएनए इन वेस्ट टाइप्स ऑफ पीसीआर। वो मैंने जब इंटरेक्शन हुआ वाइस चांसलर लोगों के साथ और आईसीएमआर के ये भी थे, डायरेक्टर भी थे, उनका था कि मेडिकल कॉलेज जो गवर्नमेंट मेडिकल कॉलेज है हम वहीं एमआरयू का हम प्रोजेक्ट देंगे। मैंने रेज किया था कि हम लोग जैसे जो प्राइवेट मेडिकल कॉलेज है वहाँ भी होना चाहिए। तो उसके लिए उन्होंने कहा कि इस साल 46 में वो प्रोजेक्ट एक पायलट के रूप में अमेरिका को उन्होंने चुना है और उसके बाद एलिजिबिलिटी आपकी होगी। तो कॉस्ट बहुत ज्यादा है मॉलिक्यूलर लैब खोलने में। बिहार में अभी एक है, अभी हमको लगता है कि एक और जगह खुल रहा है जो मेरी जानकारी है, और हम नेताजी सुभाष मेडिकल कॉलेज में खोल सके ऐसा बहुत लगेगा। गवर्नमेंट अगर मदद कर दे तो हमारे लिए बहुत प्रेस्टिजियस ये रहेगा। फार्माकोडायनामिक्स का रोल बहुत ज्यादा है। वी कैन गिव प्रेसिजन मेडिसिन, also we can practice predictive medicine, as you have given the example of pilot project done in the USA. So, with these words, I thanks all the participants, delegates, faculties, and the organizing committee for making it more successful. Thank you very much.
It is with immense pleasure and respect that we cordially invite the medical superintendent of our esteemed institution, Professor Dr. Uday Narayan Singh sir, to please take over and deliver the address.
मंच पर विराजमान हमारे एमडी, प्रिंसिपल, डीन, और ऑर्गेनाइजिंग सेक्रेटरी, and organizing chairperson, सभागार में मौजूद सभी सीनियर डॉक्टर, एचओडी, इस तरह के आयोजनों से बहुत हमारे स्टूडेंट्स को और यंग डॉक्टर्स को बहुत फायदा होता है। हम लोग ने एक आदमी का नाम तो जरूर सुना है, युवराज सिंह का, जो अर्ली डायग्नोसिस और अर्ली ट्रीटमेंट के चलते लंग कैंसर से बाहर निकल गए और उनका इलाज ये अभी भी देश की सेवा कर रहे हैं। हमें इस तरह के आयोजनों को करना चाहिए और इस तरह के आयोजन में हमेशा हमारे एमडी सहयोग देते रहे। धन्यवाद।
It is with profound anticipation and the highest reverence that we now welcome the Honorable Managing Director of our institute, Shri Krishna Murari sir, to take the dais. Sir embodies the perfect synergy of ambition and grounding. As a visionary steward of our institute, his strategic acumen and unwavering integrity have consistently propelled us towards unprecedented achievements.
Good afternoon, all. Department of Central Lab, NSMCH द्वारा आयोजित इस कार्यक्रम के गेस्ट स्पीकर्स डॉक्टर स्वरूप A.B. शाह सर फ्रॉम PD हिंदुजा हॉस्पिटल, डॉक्टर अनुराग कुमार फ्रॉम असिस्टेंट प्रोफेसर एम्स पटना, distinguished guests, participants from various medical colleges, इस संस्थान के प्रिंसिपल, मेडिकल सुपरिटेंडेंट, मेडिकल डायरेक्टर्स, डिपार्टमेंटल HODs, फैकल्टीज, UG और PG के स्टूडेंट्स, SR, JR एवं अन्य सभी, आप सभी को मेरी तरफ से हार्दिक अभिनंदन एवं स्वागत है। सबसे पहले तो मैं कहना चाहता हूं कि इस सब्जेक्ट का नॉलेज तो नहीं है, लेकिन इसका एक समझ तो जरूर है कि आजकल मेडिकल फील्ड में नई तकनीक, नई शोध, नया नॉलेज हमेशा चेंज होते रहता है, आते रहता है, और उसको अपडेट, अपने आप को अपडेट रखने के लिए इस तरह का कार्यक्रम बहुत जरूरी है। तो मैं धन्यवाद देना चाहता हूं अपने कॉलेज की तरफ से, सोसाइटी की तरफ से, गेस्ट स्पीकर्स को कि अपना कीमती समय यहाँ पे आके दिए, और आई एम वेरी मच श्योर कि स्टूडेंट्स, फैकल्टीज, पार्टिसिपेंट आप लोग कहीं न कहीं बेनिफिटेड होंगे इससे। मैं एक बार पुनः धन्यवाद देना चाहता हूं इस ऑर्गेनाइजेशन कमिटी के चेयरपर्सन डॉक्टर अनीता सुनेल मैम, ऑर्गेनाइजिंग चेयरपर्सन डॉक्टर राजेंद्र कुमार, ऑर्गेनाइजिंग सेक्रेटरी डॉक्टर स्वर्णिमा सिंह, को ऑर्गेनाइजिंग सेक्रेटरी डॉक्टर अरुण सिन्हा, को ऑर्गेनाइजिंग सेक्रेटरी डॉक्टर आरपी सिंह, साइंटिफिक कमिटी डॉक्टर विनय कुमार, साइंटिफिक कमिटी डॉक्टर ममता सिंह, साइंटिफिक कमिटी डॉक्टर शैलता ऋषि एवं यहाँ के स्टूडेंट्स को बहुत बहुत धन्यवाद और शुभकामनाएं इतना बढ़िया प्रोग्राम आयोजित करने के लिए। और इस कार्यक्रम से एकेडमिक इन्वायरमेंट तो इनहेंस होता ही है, अपना भी बेनिफिट होता है, स्पेशली हम बच्चों के लिए बोल रहे हैं, और फैकल्टी जो है इससे कंसर्न कि नए नए चीजें सीखने को मिलती है, और इनडायरेक्ट रूप से और डायरेक्ट रूप से अपने बेनिफिट के साथ साथ पेशेंट का भी बेनिफिट होता है। इन्हीं शब्दों के साथ बहुत बहुत धन्यवाद।
This opportunity to be blessed by such magnanimous dignitaries has been the providence of a lifetime. We are overwhelmed with respect and admiration in our hearts.
Respect and admiration that we would love to show and share. Thus, it is a formal request to our Managing Director, Shri Krishna Murari Sir, to felicitate our honorable guest, Dr. Anurag Kumar Sir. Again request our respected Managing Director to please felicitate our distinguished guest, Dr. Swaroop A.B. Shah Sir.
We now request the principal of our institute, Professor Dr. Hariyat Dixit sir, to felicitate our intellectual managing director, Shri Krishna Murari. We again request our respectable Managing Director to felicitate our Principal, Professor Dr. Harriet Dixon.
Now I'd like to call upon the stage Dr. Ayan Banerjee sir, who is the additional professor biochemistry at AIMS Patna, to please felicitate the medical superintendent of our institute, Dr. Uday Narayan Singh sir. I would now like to call upon the stage the Emeritus Professor Dr. Shahab Raza sir, who is also the patron of our summit, to please felicitate the Dean of our college, Professor Dr. Prem Kumar sir.
I would now like to call upon the stage Dr. Tripti Sinha ma'am to felicitate our Emeritus Professor of Pathology, Dr. Shahab Raza sir. Please, sir.
Yes. Maybe
I would now like to invite Dr. Shivendra sir to please felicitate the organizing chairperson of this summit, Professor Dr. Rajendra Kumar sir. I would now like to request our Honorable Managing Director, Shri Krishna Murari sir, to felicitate the organizing secretary of this summit, Dr. Swarnima Singh ma'am.
Now I would like to call upon the stage Dr. Ishtiaq Ahmed sir to felicitate the co-organizing secretary of this summit, Dr. Arun Sinha sir. would like to call upon the stage Dr. Animesh Gupta sir to felicitate the co-organizing secretary of this summit, Dr. Narendra Pratap Singh sir.
I would now like to invite Dr. B.V. Singh sir to please felicitate Dr. Vinay Kumar sir, who is also the scientific committee member of this summit. I would like to call upon the stage Dr. Shweta Jha ma'am to please felicitate the scientific committee member Dr. Mamta Singh ma'am.
I again request Professor Dr. Shweta Jha to felicitate Dr. Sharita Prishi ma'am, member scientific committee. Distinguished guests, esteemed dignitaries, and dedicated participants, with all honors bestowed, we formally draw the curtains on this morning's inauguration ceremony of the Bihar Molecular Diagnostic Excellence Summit 2025. We thank every dignitary for elevating this occasion with your esteemed presence. As we conclude, we firmly commit to cultivating deeper academic collaborations moving forward.
We now transition immediately to the lunch. The lunch is being served in the designated area and we invite all of you to please enjoy the hospitality. Our first academic activity will be the poster evaluation convening at 2 p.m. sharp at B block second floor. All the participants of the poster competition are requested to report by 1:45 p.m. Thank you so much.
We gather for poster evaluation half an hour after lunch. This pivotal evaluation will be overseen by our distinguished panel of judges, Dr. Bihta Shushlendi, Dr. Preetam Prakash, and Dr. Ashwin. We thank them for this critical expertise. Following poster evaluation, we will move to our highly anticipated panel discussion on overcoming technical, regulatory, and operational barriers in establishing molecular diagnosis laboratory. The venue for lunch is second floor near Central Library. Surprisingly, the venue for poster presentation is also near the same. Second floor, B block.
Thank you so much to all the dignitaries on the dais for gracing this occasion with their presence.
Thank you.